Good morning, children. We will continue with the chapter Molecular Basis of Inheritance, and in that chapter today we will start with the new topic, that is translation. The process by which the triplet base sequence, that is codon, on mRNA guides the linking of a specific sequence of amino acids to form a polypeptide on ribosomes is known as translation. There are different machineries used for the process of translation. So let us study about translation machinery. Translation requires a machinery which consists of ribosome, mRNA, tRNAs, amino-acyl tRNA synthetase, that is, enzyme that helps in combining amino acid to particular tRNA and amino acids. So let us study about the initiator tRNA. It is a specific tRNA for the process of initiation, and there are no tRNAs for stop codons. The second one is ribosomes. It is responsible for protein synthesis. It consists of structural RNAs and around 80 different proteins. Ribosome exists as two subunits in its inactive stage. Small subunit and the second one is the large subunit. Let us talk about the small subunit. When the small subunit encounters an mRNA, translation of mRNA to protein begins. Large subunit consists of two sites where amino acids can bind to and be close to each other for the formation of a peptide bond. Ribosome also acts as a catalyst. 23S rRNA in bacteria is the enzyme ribozyme for peptide bond formation. Now, let us study about the translation unit. It is the sequence of RNA flanked by the start codon, that is AUG, and the stop codon in mRNA. It codes for a polypeptide that has to be produced. Now, untranslated regions. That is UTR. These are some additional sequences in an mRNA that are not translated. They are present at both the ends, that is at 5' end before start codon and at 3' end after stop codon. They improve the efficiency of translation process. Now, let us study about the stages of protein synthesis. Synthesis of proteins takes place through the 3 stages. That is, number 1, initiation. Number 2, elongation. And number 3, termination. Let us talk about initiation first. The first step, that is initiation. In prokaryotes, initiation requires the ribosome, that is large and small subunits, the mRNA, initiation tRNA, and 3 initiation factors which are denoted as IFS, that means initiation factors. For initiation to take place, the ribosome first binds to mRNA at the start codon, that is AUG, that is recognized only by the initiator tRNA. Then activation of amino acid. The formation of peptide bond requires energy, and in first phase the amino acids are activated in the presence of ATP and linked to their cognate tRNA by a process known as charging of tRNA or aminoacylation of tRNA. In the presence of ATP and Mg+2, amino acids become activated by binding with aminoacyl tRNA synthetase enzyme. The amino acids AMP enzyme or amino acid AMP enzyme complex is called an activated amino acid. The steps of chain initiation are: the small ribosomal subunit attaches to 5' end of mRNA to form a complex. Downstream to this mRNA lies the AUG initiator codon, which initiates the translation process. Third one, the initiation tRNA joins the initiation codon by its anticodon through hydrogen bonds. The fourth step is now the binding of large subunit of ribosome with its small subunit completes the translation initiation complex. The fifth step, at this stage, the initiator tRNA lies at the P site, aminoacyl tRNA site of the ribosome, and the A site, peptidyl tRNA site, is vacant to let another charged tRNA to enter. So that is all about the initiation of protein synthesis, the first step. Now let us study about elongation of polypeptide chain. In this step, another charged aminoacyl tRNA complex binds to the A site of the ribosome. A peptide bond forms between carboxyl group, that is COOH group of amino acid at P site, and amino group, that is NH3 group of amino acid at A site by the enzyme peptidyl transferase. During this stage, ribosome moves from one codon to another codon along the mRNA in the 5' to 3' direction. Amino acids are then added one by one in the sequence of codons and translated into a polypeptide sequences dictated by DNA and represented by mRNA. That is all about the elongation process. Now the third step of translation is termination of polypeptide. When the A site of ribosome reaches a termination codon, then no tRNA binds to the A site of ribosome. At the end, a release factor binds to the stop codon and translation is terminated, thereby releasing the complete polypeptide from the ribosome. This is the picture showing the process of translation. In the first diagram, here, the initiation complex forms when the ribosomal subunits and the first tRNA molecule lock onto a strand of mRNA at the start codon, that is AUG. A tRNA carrying the amino acid specified second in the mRNA sequence plugs into the complex at the A site. Then, a peptide bond forms between the adjacent amino acids. You can see here red color peptide bond is formed between the adjacent amino acids. As the first tRNA detaches from the mRNA template, the second moves over the P site. You can see this has got removed. So second will move to the P site, trailing its small amino acid chain. A third tRNA sites down at the vacated A site, which is now situated over the next 3 letters of the sequence. And in this way, a complete polypeptide chain is formed. So children, that is all for today. In this video, we have learned that how the mRNA information gets translated and produces a polypeptide chain, which is nothing but the proteins. I hope you have understood this topic. In our next video, we'll be learning about operon. Thank you.